Diagram, and I want to make sure that you guys had an idea of how this DNA works. This is not a super simplified version, but it's a little bit of a blown up version, and I wanted to see the exploded version on the bottom because when we get into the DNA, what we're doing today is DNA replication. You've got the basics of it. You had the basics of it in 5th, 6th grade or whatever you had. And it's the parts, maybe not what a nucleotide is, but you should have learned A goes with T, G goes with C, shape and such like that, okay? But when we get into this replication thing, what does replication mean?
Duplicate, like
Make copies. So we need to make a copy of it in order to make new cells. That's the only reason we're making copies. The other thing that was on the notes that you had yesterday, DNA, even though we said if you string it out, you have the 6-foot long piece in every nucleus. I said it's all spread out within the nucleus itself. There's all kinds of different spots, different genes within that DNA strand or different strands that are going to be opened and closed at different times. So you want to have that hydrogen bonding, and it showed it on here. The hydrogen bonding is it's a really weak chemical bond that can be broken easily and put back together again. So we're going to look at it from, hey, we got to open this thing up because you don't read it when it's intact. You open it up, and on one side it may have the directions for one thing, and on the other side it may have something completely different. So the cell knows which side to read. But you want to be able to open it up, and as soon as you open it up and you read it, it almost immediately they close back up. And this is when we talked about enzymes before. One of the enzymes do a lot of this work. There's a specialized enzyme for opening it up, there's another one for closing it, there's another one for reading it, and it just goes on and on and on. So I wanted you guys to know that you have this weak bond, it holds it together, it opens it, reads it, puts it right back together again. The other thing is when you read it, this is the kind of goofy part, it's read in a direction, okay? So if you go on the bottom, I want you to see this. Don't get too caught up on this, but it is kind of important. I wanted you to see this, and this is one of these chemical things that you get into if you get into chemistry and such. They talked about the sugars being rings, either a double ring or a single ring. You have like the single ring right here, you have a double ring right here. Okay? Let me focus this a little bit more. Okay. Do you see where it says 3' and 5' at the bottom? Circle those things where it says 3' end, 5' end. And I had a couple of teachers that were teaching here before, and they saw this, and I said, hey, and we mentioned it, and they're like, oh, I didn't really know what those are for. And it's like, no, there's a really specific purpose for that denotation, that numeration on there. Follow this. When you look at the sugar that's attached to the nitrogen, okay, we'll start with this one right here. Wherever the nitrogen's attached, right here, okay, you have carbons that make up the basis of that sugar. So we'll start off with this. This will be the #1 sugar. So you got, carbon. This will be 1 carbon, 2 carbon, 3 carbon, 4 carbon, and then there's a 5th carbon down here. Okay, so the 5th carbon is down here, hence they call it, this is the 5' end. The prime, I'm not really sure where they come up with that. What it means is that this is your 5th carbon. Okay, this is your 5th carbon. That's essentially pointing down. Right? Now if you go to the other side, and do the same thing, you go, here's a carbon here, this is your #1 carbon here, and you start counting around to 3. Which one's pointing down? The 3 is on the bottom on this side. So you have the 3'. Okay? So the important part about this is when the DNA is actually either read or duplicated or replicated or whatever, it has to be read in a certain fashion. Okay? So it always is read in a 5' to 3' direction. All right? So on this side, because this is the 5' end, I want you to write this. 5' end, we're going to read in this direction to the 3' side. Because if you do the same thing and you do the counting on this molecule right here, this is the 5', this is 4, what's this one?
3.
That's 3, that's the 3' end. So we're going to read from 5' end to the 3' end. So the enzymes that attach this to start reading, they'll attach right here and start working their way up. We're still doing the same thing, it's 5' to 3'. If this is 3', what's this? 4. What's this? 5. So on this side, we're going to go 5' and still read 5' to 3'. We're going to read in this direction. So the weird thing is, if you can think about it like this, we're going to read, this makes sense to read in this direction. We're going to start where it's unzipped and keep reading down, reading down, reading down. This is kind of a backwards thing. So when you think about the letters kind of being read, almost think about these guys are upside down. Whereas we can say this is your G right here. So you have G, I don't remember this. We'll say this is T up here. This one's C, right? So think about C as kind of like being upside down. If this is T, what goes over here? Think about that kind of be upside down. and it's reading in that direction. That's all that means. The only important part of this is really that you read from a 5' to a 3' direction. Okay? And that'll come into play a little bit later. Okay? On the notes, the sheet of paper you have, take out a sheet of paper, one blank sheet of paper. You should have had that ready. You can put the coloring sheet aside. We really don't need that. I'm going to use this board. I like to do this, especially if we take notes and we can do it in like a drawing sense. I'm going to use this board right here. So however much room you need, you have some colored pencils, use them if you like. If you just want to do pencils, just regular pencil, it's fine too. But I'm going to use pretty much this whole board. Okay? I'm going to look back and forth at my notes a little bit here and there, but we should be able to get caught up in this. The one thing I want you to put in the corner is the replication. Well, where's the DNA at? Always the nucleus. So replication happens in the nucleus, and DNA replication is a way of making a copy of the DNA so we can make new cells. So we'll start up here, put DNA replication. And this was part of your learning target for today. Know how DNA replication works and what the purpose is. So to make exact copy of DNA to make new cells. Bonus. You guys are all going to learn this process. The name of the process by which cells copy themselves or make new cells. Starts with an M. Mitosis. That'll be our next stop. As soon as we get done with replication, we're going on to mitosis. We're going to learn the specifics of that. So before we get to mitosis, we want to make sure we make a copy of the DNA so we have the base instructions to make those new cells. Okay? The other thing is this. If we're going to build new DNA, we need to parse the new DNA. So off to the side, just way off in the corner here somewhere, just so we remember, let's draw a little cell. Make a nucleus in there. Not quite that big, but that's good. Put a little double helix DNA. That's horrible DNA. And like we talked about before, this is one of those really difficult things for people to get. Every cell has blood flow to it. You get contact from the outside world and to every other cell within your body through this blood flow. So I'm going to draw a little blood vessel going here, going along here. So, I'll wait till you're ready. You don't have to use whatever colors you want. DNA is made of repeating patterns of what? Nucleotides. What makes up the nucleotide? Sugar, phosphate, and nitrogen base. So when you guys looked at strawberry and you had the DNA, as soon as you eat all that DNA, you use carbohydrates for, what do we use carbohydrates for? Energy. Lipids?
Fat.
I know it's fat. Hormones. Stored energy, worse, you want to use it later. Carb is first. Mainly you're using for hormones. Proteins you use for building more proteins, okay? But we said nucleic acids you do not use for energy. That's your 4 molecules. So if you take in all this DNA, what do you do with it? You break it up into its respective nucleotides. So I always imagine this, floating among all the other stuff that's in your blood flow from the nutrients you take, just make yourself a little like phosphorus, attached to deoxyribose, attached to a nucleotide. And you have these, what they call free nucleotides. Squeeze it on the edge. Free nucleotides. Meaning this, they're not bound up in any DNA structure. They're the ones that you broke down in your diet. You ate those strawberries, you ate whatever, you're breaking down this DNA. Sometimes it's already broken up just by processing it, by the packaging and grinding wheat and all that other stuff. But you're still taking it in. Now your body's going to use that because that's going to go into, your body's going to use those free nucleotides. Active transport's going to pull it in. They'll be in the cell, and they're just roaming around the cell. When the cell goes through DNA replication, though, it can use those free nucleotides to start building the rungs of the new ladder. Okay. Now, are we caught up? Okay. So let's kind of like break it down, and expand it, and this will be one of those things we'll go over a lot just to make sure you got it. How many pairs of chromosomes do we have? 23. We have 23 pairs of chromosomes. And what did we really say chromosomes are?
DNA.
It's all just DNA. Okay, it's just DNA. So you get a set from mom, a little symbol like that. Set from dad. Did we talk about how you can remember this is dad? What does it look like?
A penis.
Looks like a penis. That's the easiest way to remember it. It is, for real. Okay, so you get 23 pairs. So you get one set from mom, one set from dad, and they're complementary pairs. So they look, when they're bound up just before mitosis, they have this kind of like the exclamation point kind of over top each other. So you have one there, you have one there. They're supposed to look identical. Do the best you can. So we'll call this is the number 1 chromosome. This is also the number 1 chromosome. And you continue, I'll put little dots, 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 dots. So it knows that we're cutting through all of the chromosomes until we get to number 23. And the 23rd pair is going to be the X or Y chromosome, depending on if you're a boy or a girl. Okay?
So those dots are basically the...
Yeah, just the other numbers. So 2, 3, 4, 5, 6, 7, all the way until you get to the 23rd. Okay? Make sense?
What about for Down syndrome?
It's a totally different thing. We'll talk about that later. For Down syndrome, depending on which side on one of the sides you have an extra copy, you'd have 2 21s. You'd have 2 number 21s. Okay? Let's take one of these chromosomes. Just random, it doesn't matter which one, just pick one. Right? Take one chromosome. And we said that basically these chromosomes, I'm going to make a little notation up here. You may already have this, so you don't really need it. Chromosomes are just condensed DNA. And we said if you take the DNA and you wrap it on itself, and then you take that and you wrap it on itself again, and you wrap it on itself again, kind of like a telephone cord, you triple wrap it, that's what makes up chromosome. I'll show you a picture of it later. They end up wrapping on these little protein balls called histones. But it's all just basically DNA. Now if you take one section of that, and this is what they do, they call it gene mapping, or they have gene markers. Genes make up the smaller part of chromosomes, and then that works down the DNA. They have these little markers right here. There'll be one here, there'll be one here, maybe there's a fat one right here, one's here. And they've mapped up each of the different chromosomes to say, here is where this gene lies. Here's another gene. Here's another gene. You can have several different markers all over it. It doesn't really matter. But they give them different names too, and they usually control, they control something different. They control a trait or multiple traits, or their conjunction with another chromosome. Okay? So these little sections, these are our genes. Okay? And a gene is just a section of DNA that codes for a trait. I'm going to put trait in quotations because in your reading yesterday I talked about it could be a morphological trait, a structural one, behavioral, it could be physiological, whatever. So you have the codes for different traits. All right. Usually, thousands of base pairs long. Each gene usually has thousands of these base pairs. So when you look at that double helix, it just keeps going and going and going and going and going and going and going. When you're done, put your pencil down. Okay, let's expand this and blow it up a little bit. It's kind of like we did on the coloring sheet. I'm going to try and take like this section right here. And I'm going to make a little double helix. That's our condensed DNA. And then I'm going to take it one more step. I'm going to use this little like expansion bar where, here I'll do this, kind of blow it up in this direction so we can see a little bit better. And then I'm going to make, I'm going to go like this and try and make one. Thing of DNA. There's actually a number of base pairs that are each twist. For 6. It's off a little bit. That's okay.
Wait, how many do you need?
We're going to do 6 on this side and 6 on this side, little brackets coming off. Like I said, I'm just trying to utilize just this piece of board right here, so I shouldn't get any bigger, but I will jam it full. Okay, so we got the DNA. We got it from the chromosomes. We looked at one gene, makes up DNA. And the purpose of all this is to look at this DNA replication. Okay, so how does the replication work and what takes place? All we're doing is making a copy. How are we going to do that? You see it's unzipped. We've broken the hydrogen bonds. The way that you do it is, when it's together like this, I'm going to draw this. There's an enzyme. Enzymes are usually gigantic proteins. Enzyme that's going to read and open up. Enzyme unzips.
Why did you put a circle there? Like
That is, the circle is the enzyme.
oh, so
I can't make a really good enzyme. They're really peculiar shapes.
oh, so it like runs along it. And like,
It runs along it and unzips it all the way up. Okay, so I'll put an enzyme in there, and I'm going to show it unzipping. All right. So as it unzips, I'm going to move the enzyme in a bigger picture right here. Here's my enzyme. And they all have particular names depending on what their job is. This one I believe is called. Well, they have one called DNA helicase. ASE on the end means enzyme. So helicase is going to unwrap the helix. That's its job. And then they've got other ones called DNA polymerases. Polymer means that you're building a molecule. So the other molecules, they have one will go on one side, one will go the other side, and they start reading the DNA and attaching free nucleotides. Okay. Now let's do this. We'll say that this end. We'll say that this end is 5 prime. So what end is this?
3.
This is 3 prime. If this is 5 prime, what does this have to be?
3 prime.
This has to be 3 prime. So what does this have to be?
5 prime.
5 prime. Okay.
and it's the top one.
This will be basically top. There's no top or bottom. There's no. It's a matter of reading. For your understanding, just think of it. That's what it's doing. Is it's reading it in the backwards direction. But there's no top and bottom in DNA.
Okay.
There really isn't. We just know this. Which direction do we always read?
5 to 3.
5 to 3. So I'm going to write a little arrow. I'm going to put read on here. I'm going to draw an arrow this way. And I'm going to write read here. And I'm going to read that DNA to see what it says in those directions. Can you guys read okay back there?
Yeah.
Okay. Now, here's a couple like goofy terminology things, and you don't need to have particular colored pencils for this, but we do got to get this. There's the 2 different strands. They call it, this one is called, this side we'll call it could be either side, but we're going to designate this one. This side we'll call is the original. They're both parent strands. They're both parent strands. This is a parent strand and parent strand. So we're going to call this the original parent strand. The other side would be the complement. And it's when you copy stuff, you do the complement of it. If this side's A, this side's going to be T, right? And you're not copying it, but you're reading and determining what the complement of it is. So this side, and I'll just try and point an arrow out here. This one's called the complementary parent strand. I'm going to move these just a little bit. The other thing that you have too is when you build the new strands, they're going to be called daughters, but the point at which you build it, this spot, this opening, is called a replication. Replication fork. You split it open, that's where you start to replicate it, so they call it the replication fork. Fork, yeah? Fork.
It's
Just
just
like
pointing
a spoon or a fork. This spot, this opening is called the replication fork. Is that okay? The one other weird thing about it, it doesn't really mean much now, but it will mean much. It'll mean a lot when we get to the protein synthesis spot. I'm going to do this. I'm going to put this as A, T. I don't know. Pick a letter. A, T, A, T, G, C. Pick one.
G.
G.
C,
C.
C,
C. A. Awesome. All right. So what's the other side going to be?
T.
T.
A.
A.
C, G,
G,
A, T, A, T.
A. T. There we go. When you're doing the straight copying, all they're doing is reading the individual ones. When we get into protein synthesis, the proteins are made by amino acids. In order to determine the amino acid, you read these things in codons. So like this little spot right here, I'm going to try and make a little bracket. This 3 base section. So there's a 3 base section, another 3 base section, another 3 base, another 3 base. This little 3 base section, I'm going to kind of draw a little arrow out here. This is called a codon. All right. When you're done, put your pencil down.
What's a codon?
I'll tell you. I'll tell you. Well, I want you to make sure you make this distinction. A gene is a section of DNA. It could be thousands long. A codon is a 3 base section of DNA. You read them in 3 bases. So it's going to read 3 bases, the next 3 base, the next 3 base when you do the protein synthesis. We'll look at that later. For now, I just want you to be familiar with the term. All right. The next thing that you need, the enzyme plus a little bit of ATP, plus ATP, just a little bit, unzips. Then because we're reading 5' to 3', we'll have another enzyme that attaches here. Well, it attaches at the beginning, but we'll put it right there. It attaches right here and starts reading in this direction.
It's only on the one side though?
And you put one on the other side too.
Okay, so there's one enzyme per side.
Yep, there's one on that side, and you have another one on this side.
What's that? And they read 3 bases?
Now, the enzymes, they're going to read along the strand, depending on which direction we do. So up here, they're going to read 5' to 3'. On this side,
What does the big one do then? Does it just say?
that's unzipping it.
Oh.
It's unzipping it. These guys are going to read along it, and what they're going to do is these enzymes are going to attach free nucleotides. That's why I put them in the same color. So what's happening is the enzymes they're going along and they're reading one step at a time. They're taking the free nucleotides that you got from your diet. They're just floating around. They're in the cytoplasm. So because they're in the cytoplasm, they're also floating around the cytoplasm in the nucleus. And it's grabbing it. And as it reads it, I should have started this enzyme up a little bit farther. But as it read this first one, what it did is it read, oh, I have an A. So what am I going to attach? You're going to put a T right there. I read a T. What am I going to attach? I read a G. What do I got? T. Next. G. Next. And lastly, T. This guy's doing the same thing on the other side. He's reading along. He attached an A. Oh, I got an A, so I need a what? T. 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 G. G. T. A. So when you look at it, if you were to like cross this out, hold these two, whatever, this side ends up being an identical copy to this side. Alright. See it?
No, I don't do that.
So now what happens is as it's going along here, as this enzyme is going along here and it's attaching it, it's also going to suture together these individual nucleotides by attaching it with those phosphates. It's going to put a phosphate in between each one of those, or attach the phosphates to the sugars. Okay? And then as this reading through, it's also attaching these two. Now this is only one more weird thing that you gotta be able to do, and I'm going to stick it in here. These two new strands that you've connected to those original parent strands, these are called the daughter strands.
All the new ones?
The new ones are called daughter strands, yeah. So watch. This daughter, this is the daughter of the complementary parent strand. This is the daughter of the original parent strand. Now, if I could very well call this side the complementary parent strand as long as this one's the original. It doesn't really matter. But if I'm saying, hey, so what is the, read off the first codon on the daughter of the complementary parent strand, right? You go, okay, complementary parent strand's here. This is its daughter. This is the first codon. What's the first codon on the daughter of the complementary parent strand?
ATG.
ATG. Okay? And that's why we need to know some of that terminology stuff, just to say, okay, where's this, what's this, what's this? Okay? Does that make sense?
Yeah.
Okay. Here's what I want to do. I'm going to pass out this one worksheet. This is a little bit of practice. It's a little bit different. Put your name, date, period on the top. Take a look at it. Tice, keep going.
Dude, I should have made my DNA a lot bigger. That's all I said.
I'll leave it up there for a little while. Okay?
I get it now.
I'll leave it up there for a little while. Pass them out quickly. I got to show you how to do this. No, just leave it. Too much. You pass too much back. You're only supposed to pass back what you need. Pull it up. Let's go. Nothing. Yeah.
No, like John doesn't pass him before back.
Everybody's got one?
Mr.
Anybody else need one? Wow. Logan, figure out this?
Pass that
Who else needs one? Pass it back. Pass it back. Pass it back. Who else needs one? Andrew? Anybody else? Ashley? Go. I think there's two coming back, Tyler. Does everybody have one? Never had such a colossal failure. You need to pass it back. They're all waiting for you. Anybody else? Fantastic.
That's just external shoulder rotation. That's a lot.
Okay, so when you look at this, there's no easy way to do a worksheet with this type of thing. This is just something I made, and I tried to make it so that you can practice this. I'll help you a little bit with the first one because the first one's a little bit goofy in the sense that you're trying to figure out where all these spaces are, okay? I'm going to read along with me, and I'll start you off to kind of give you an idea how this works, okay? It says complete. Complete and label the following DNA sequence using the terms. You need to use all these terms: original parent strand, complementary parent strand, daughter strands, codons, '3' and 5'. Everything has to be labeled. Okay. You're going to place the original parent strand, which is TCG, GGG, ACT, CTC. Watch. Always do this. This is like it's the same thing with the cladogram. First thing you want to do with the cladogram is do what?
Draw
Label.
Trait gates, thank you. Trait gates, put in your notes every single time. First thing, when you see a DNA strand, do this. I do this because it gets too confusing. I'm going to break it into its codons. So I see my strand right here. I'm going to break it into its codons. Boom, boom, boom. There's my codons. Just so I can keep track of it.
What was that?
Now, if I don't give you the 5', 3' end, you won't know, so I'm going to have to give it to you. What we're going to do is, it says place the complementary parent strand in the last column. Here's your last column. The replication should take place in the middle 2 columns. So the middle 2 columns will be called what strands?
Daughter strands.
Daughter strands, okay? Here's what we're going to do. The first side, we'll do this. Yeah, we'll start with that. We'll put this one, this end up here is 5'. It's 5'. So we'll start building the strand in here. What are our first 3 letters? I'll put T, C, G. If this is our original parent strand, where's our complementary parent strand? Last column. So what's this first space going to be? A,
G,
G,
C.
C. Okay? And I'm going to build my daughter strands with that. Here's your only task. Complete the rest of the spaces. Your task is to make sure that you label it and you've got the right complements done and you've built the correct strands. Okay? So when you get done, I want you to do this. When you get done, make a little squiggly right here. What's the purpose of DNA replication? Not to make new cells. Make more DNA for what? More cells. I need to have 2 identical strands. So here's how you can kind of check yourself. This side will be for cell number 1. This side will be for cell number 2. But you should have identical sets when you get done with that, okay? So here's what I want you to do. Just it probably won't take you too long. When you've got it done, let me know, raise your hand. I'll move you on to the next spot, okay? Finish this up. Make sure you process it. Work with your partner. Do what you got to do. This is going to be the starting point for everything. Questions? Okay, good.